I'm Dr. Matthew White. My research started at Cardiff University a few years ago now. I was there for an undergraduate degree in biomedical science and then a PhD in developmental neuroscience. So really studying how the brain starts to form, how nerve cells in the brain grow, reach their, form the right circuits and connections and things like that. A few years after that, I moved to the Bateman Institute, which is a nice institute near Cambridge, and started researching motor neuron disease primarily. So looking at RNA binding proteins and how the control of our DNA code is important for the function of nerve cells. And a few years into that, as a group, we moved to King's College London, and we've been here ever since, setting up our research program.
Hi, so I'm Sarah Milani. I'm a PhD student. Matt is my supervisor. My research has always been kind of focused on motor neuron disease, so I'm quite passionate about it.
So really, where we feel there's a gap in our understanding is the earliest changes that occur in the human brain, and that's just mainly because of how difficult it is to access, the human brain in an individual, in an adult. But then taking into account that the brain is a incredibly complex organ of many different types of nerve cell and other glial cells that function together, different cell types can have different things going wrong at different stages of disease. So when you take all that complexity together and the fact that no two individuals are the same, so how the disease might initially begin in an individual's case of motor neuron disease and how it may progress can be different. So the project we're funded for now is to actually expand on our stem cell approach to make these kind of small mini brain cultures, so like 3D pieces of neuronal tissue that represent regions of the brain. They can contain quite a nice mixture of different types of nerve cell and other cells of the brain that advance on our previous kind of two-dimensional cultures, and then to really study why certain cell types are vulnerable and what we can potentially do to overcome those vulnerabilities, crucially at the earliest stages of disease onset. Human stem cells creating, human mini brains. They're very small. You're talking a few millimeters in size, and they can, grow slightly over the course of several months. So we're not talking about large structures here, but they can contain, upwards of a million cells, different types of neuron, and as they get a bit older, other types of cell that are important to the function of the brain beyond just nerve cells. So we're trying to, if you like, understand in certain cells in the human brain at the earliest stages of the onset of motor neuron disease, what's going on, for two real reasons. Can we use some of those signatures we uncover to take forward and be able to better understand in individuals what's going on at the start of motor neuron disease? But equally, if we can then try and correct some of those signatures we spot, can we apply them back to the appropriate individual? I think hopefully in the long run, that kind of strategy will help improve clinical trial, where we can get the appropriate new therapeutic to the group of patients. We have the best chance of finding those new therapies that could make a difference. And ultimately one day, can we spot in an individual as early as possible what's going on and target them with the correct, more personalized therapy? Motor neuron disease is incredibly complicated. More of an umbrella term really of potentially different pathological processes that are going on. No two individuals are the same. So that kind of approach, I think, is where we could have greater impact for people. The catalyst is fantastic. I suppose it's to do exactly what it maybe says on the tin, is to really get our new systems off the ground, accelerate the early progress, so we can get the mini brains established, running, specifically in our MND context, as quickly as possible, so that we can scale it up in the future. If we can get into the early stages of the disease process in cells from a human genetic background, I think we can make some interesting observations. there's a couple of key areas where I think if we can make advances, it could have more impact for people. It's one thing understanding what's happening at the later stages of disease, as the process is, if you like, well underway. It's another to understand what the earliest changes are. Can we actually, in individuals early on, potentially even before they develop symptoms, spot signs of early disease process? And can we design therapies that could potentially correct some of those risks? And that's important for how we do clinical trials, how we develop new therapies, but ultimately one day how we apply the correct therapy to the correct individual in the clinic as early as possible.